Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A paper from 2020. So which of the following human diseases is caused by fungus? Athlete's foot is, influenza is a virus, malaria is a protoctist or protista, and then tuberculosis is a bacteria, so it's A. Which of the following plant diseases A to D is caused by a pathogen from the kingdom Protoctista? Sublime bananas is caused by the fungus Mycophylla aeruginosus. I don't even pronounce that, but it's the fungus, and that's out. Ring rot is a bacterial infection of potatoes, so that's out. Tobacco mosaic disease is due to a virus, and tomato blade blight is the only one that is, so it's D. This is caused by a protoctist with the name Phytophthora infestans, and it's kind of like it's almost like a fungus in the way it behaves, and it penetrates the cells, destroying leaves, tubers, and fruit. And yeah, which of the following statements A to D is not true of human erythrocytes? Erythro means red, cyte means cell. So they're produced from stem cells. Yes, they are. They're created from bone marrow stem cells, the hematopoietic stem cells. So that is indeed correct, but they want not true. So that's out. They're made in the bone marrow. Yes, they are. They are specialized cells. Yes, they are. They undergo mitosis. No, they do not. Because erythrocytes eventually just get rid of practically everything and are just hemoglobin carriers. So when they've reached that point where they've specialized, they can't undergo mitosis any further. They're just doing the job. And then they eventually get destroyed in the liver or spleen. So questions 4 and 5 refer to the following image of a human blood smear. Which ones of A to D show a lymphocyte? So we're looking for a cell in which it's got a singular big fatty nucleus like that, which is D. So the cell labeled E shows a parasite, trypanosoma. Which of the following statements is evidence that trypanosoma is a eukaryote? So if we look at E, where are we? There it is, right? So then it says the nucleus is its nucleus is present. So although it's not very clear in the image, it's asking you what this state provides evidence that it is a eukaryote. So that's going to give it eukaryotic-like properties, because eukaryotes have a nucleus, prokaryotes do not. It is a similar size to blood cells. Prokaryotes are a lot smaller, so the eukaryotic cells tend to be bigger and more complex. That's true. And the presence of a flagella doesn't guarantee that it's a eukaryote, because prokaryotes like bacteria can have a flagella as well. So it's just 1 and 2, which is B. Plants can produce a variety of chemicals in response to pathogens. Which of the following A to D is produced by plants in response to pathogens? So they can make insect repellents, insecticides, antifungal compounds, all kinds of stuff like that. And antibacterial compounds are indeed one of them, including antibiotics like phenols. So in that case, A is definitely true. Now, antibodies are made by B cells. These do not have B cells, so that's out. Ethylene promotes ripening of fruit, so that's... Penicillin is made by fungus, so that's out. So it's A. Scientists self-pollinated some pea plants that were heterozygous for the gene controlling height. They expected a 3-to-1 ratio of tall to short plants in the offspring. 1,046 plants grew in the next generation. 798 were tall and 248 were short. Which of the following A to D is a statistical test that could determine if these numbers are significantly different from a 3-to-1 ratio? So they're expecting a 3-to-1 ratio, and they got these actual numbers. So they want to see how much different what they actually saw was different, to the ratio they expected. So when you're comparing to see if there's a significant difference in the expected and observed results, the statistical test you would do is the chi-squared test, which is A. You don't do the Spearman's rank, because that's looking at correlation between two things, and to see if the correlation is significant. Standard deviation gives you the spread of results about the mean, and the t-test is comparing the differences in two means and whether they're significant or not. So by that, it's got to be A. So polar bears, Ursus maritimus, and giant pandas, Aloperoda thingy-majiggy, both belong to the family Ursidae. Which of the following is not true about the classification of polar bears and giant pandas? Desperate King Philip cried out for goodness sake. So domain, kingdom, phylum, class, order, family, genus, species. So if they're in the same family, then they've also probably they're going to share all of these all the way up to that point. So every single one of those is shared. It's these two that are different. So whichever one of these is just, not keeping with that is the ones that are going to be not true. So they both belong to a different class. Well, they don't, because if they in the same family, they must be in these two. So that's immediately in. They both belong to a different species. That is true, because you can see here there's species name, the genus and species are different. If they both each belong to the same order, Carnivora, again, that's coming before family, so that's true in this case, but they want not true. They each belong to the same phylum, Chordata, again, same thing. So in that case, it's got to be A. These are, as you go down it, it's a hierarchy of things. So like, for example, humans are from Hominidae, and I think we share that with monkeys. So up to that point, we're exactly the same in terms of these things. Which of the following statements are that DNA replication is semi-conservative? So after one replication, the number of adenine nucleotides is equal to the number of guanine nucleotides. Well, no, because they're not complementary base pairs as well. So that one's out. After 2 replications, 2 DNA molecules have 1 original, 1 new strand. and 2 have 2 new strands. And that kind of makes sense because if you think about it, here's the first, right? They replicate, one acts as a template, and then a new one comes along. And then from these, again, one acts as a template, and the other one acts as a new template, like that. So after 2 replications, 2 DNA have the 1 original and new strand, and 2 have 2 new strands. So that is indeed correct. After 3 replications, there are 8 DNA molecules, of which 2 have the original. Yes, the original DNA, because if I do that again and with the next replication, there will be 8 of them. Each time you do a replication, you're timesing by 2. So if I'm doing 3 replications, it's going to be 2 to the 3, which is going to be 8 strands as well. And they only have 1 copy of the original, so that means it's 2 and 3, which is C. Which of the following reactions A to D describes the conversion of a polymer to a monomer? So immediately I can already see esterification is, combining these, are condensations combining those, so that's out. Now we need to look at these other ones. We're looking for a polymer to a monomer. So polymer is 3 or more, whatever it is, monomers joined together. Maltose is only 2, so that's out, so it's got to be D. Which of the following A to D is true of a competitive enzyme inhibitor? So it binds to a site other than the active site, no, that's a non-competitive. It can bind irreversibly at active site, no, that's non-competitive. It changes the shape of the active site, no, that's non-competitive. Effects can be overcome by adding more substrate, that is indeed D. Because if you think about it, reactions in biology require an enzyme to collide with a substrate and lead to reaction. Now, if we have an inhibitor, for example, that's the inhibitor, this is the substrate, yes, the competitive inhibitor can bind here and then just detach. But if you have like this one inhibitor amongst a bajillion, of these, like substrates, then the chances of it hitting and colliding and leading to reaction with substrate is way higher than an inhibitor. So you can just add loads and loads of substrate and it will overcome that and you'll eventually get back up like that. So which of the following A to D is not true about adult stem cells? They are found in the bone marrow. Well, I mean, you've got your, again, hematopoietic stem cells, which make all of your blood cells. So they are in the bone marrow, yes. They are not specialized. That is true because they then become specialized. They are totipotent. That is the one that's untrue because totipotent means it can become literally any tissue type in existence. I mean, obviously what, it can make. So that one's already out. They can be used as a renewing source of undifferentiated cells. Well, I mean, that is true because again, red blood cells and all the other sort of things that it comes down from. So your blood cells and stuff, they can all come from there. So that one's out as well. So it's just C. The nitrogen cycle involves a range of reactions in microorganisms. Which of the following processes A to D usually undergoes anaerobic conditions? So the nitrogen cycle involves a range of reactions in microorganisms. Which of the following processes A to D usually undergoes under anaerobic conditions? So the two that do that are denitrification and nitrogen fixation. So, I mean, that's already D here. We'll talk about the others, though. So, conversion of amino acids to ammonium compounds is what your liver does when you're deaminating things, so that one's out. Conversion of urea to ammonium compounds, there isn't really a process that does that because that happens in your body. We want to convert these ammonium compounds into urea because it's less toxic, so that one's out. Nitrification is just simply where you're converting ammonium ions into nitrites and nitrates, and that's not anaerobic conditions. So it's only nitrogen fixation. That's a bit beyond A level, but it helps me understand this, is that nitrogen fixation and denitrification both happen in anaerobic conditions. Reason being is for nitrogen fixation, there's an enzyme that they use called nitrogenase, and when you have lots of oxygen there, the nitrogenase essentially just breaks down. And for denitrification, what happens is, when you have anaerobic conditions, the bacteria switch to using nitrite, NO2, instead of oxygen as the electron acceptor in respiration. So that's kind of how I remember them, just to make sense of it, because it's just a bit, weird. Which of the following bacteria converts ammonium compounds to nitrites? Nitrification is when ammonium ions, NH4+, become nitrites, and then they are switched into nitrates. So the ones that are changing into nitrites are called nitrosomonas. And the ones that make it nitrates are called nitrobacter. So in this case, it's nitrosomonas. It's not azotobacter because that's involved in nitrogen fixation, which is where you're converting, atmospheric nitrogen into ammonia. It's not nitrobacter just because I said it converts it to nitrates. And it's not rhizobium because that's also to do with nitrogen fixation, so that's out. So now it says ash trees are common throughout the UK. They often grow in dense woodland. Which of the following A to D is an abiotic factor that's likely affecting the growth of young ash trees? So the availability of light underneath larger trees in the wood, well, definitely, because that would affect photosynthesis and the creation of biomass, the conversion of things into biomass and stuff. So that's definitely one. The availability of oxygen in the air, not necessarily, because they use CO2 and water and sunlight to make glucose. So not as massively. The presence of a pathogen that causes, well, that's not an abiotic factor. And the species of bacteria in the soil, again, not an abiotic factor. So the main big one really is probably going to be A. So the body plan of multicellular organisms is under genetic control. Complete the passage below using the most appropriate terms from the list. So body plan, remember, is just the general structure of an organism. It's just how all the things are arranged in a particular way. And this is to do all with controlling the expression of genes and proteins and stuff. So the development of body plans in eukaryotic organisms is controlled by, we need to figure out what specific genes they are. And if we narrow it down, we have something called Hox genes, and within those Hox genes, you've got these regions. Here's a Hox gene. Within them, you've got regions, which are called homeobox sequences. These homeobox sequences will make a part of a protein called the homeodomain, which then goes and binds to specific parts of DNA, right? And it enables this protein, that homeodomain protein, to work as a transcription factor. It binds at the start of, genes that are involved in, developing the body plan, and it can either activate or repress the production of proteins involved in making the body plan. So here then, it would be controlled by homeobox genes. These genes code for proteins that are able to bind to, well, as I just said there, DNA, and turn specific genes on and off, and are known as something that can affect the transcription, because when you turn on a gene, you're transcripting it and translating it. So you transcribe it into mRNA, you translate it to ribosomes. So if you're able to switch on and off, you're affecting its transcription. So these are transcription factors. These proteins contain a sequence of base pairs that vary little between species within the animal, something, or fungus. So the word that would fit here is going to be plant, and then or fungus. Well, these are all kingdoms. And yeah, these homeobox sequences are highly conserved. What that means is they're not changed very much throughout evolution in these different organisms that have these homeobox sequences. Investigations into the activity of genes that control the body plan frequently use fruit flies in mice. One reason that fruit flies use is that there's less public concerns about the ethics of using flies. What's two of the reasons why fruit flies are chosen for research contributing to the development of body plans? Well, I mean, first of all, they can rapidly reproduce, which means you can, just use a lot of them quite fast. And you can look at more generations in a given time. Because, you don't want to spend ages waiting for them to, reproduce and pass on to this and that, blah, 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 whatever I'm trying to look at here. And as well as that, they also have simple body plans, right? You don't have a really complex organism and just all of this kind of stuff. Other things you could say, low cost, and other things as well. For example, we already know quite a good amount about fruit flies as well. Their genetics are quite well understood. There are some public concerns about the ethics of using mice in these investigations. Suggest two reasons why mice are chosen as a suitable species for an investigation. I mean, I guess another thing as well is, again, it's low cost and rapid reproduction rate. And you can also say, for example, their genetics are quite well understood. But I think another stronger point is that they are more similar to humans, right? Like, if you're going to do research in flies and stuff and then try and translate that to humans, it's a lot harder to make that more relatable versus, like, a mouse. So tannase is an enzyme produced by some microorganisms. It's useful in many industrial applications, including food. The tannase used in food production can be free in solution or immobilized. State one method by which tannase can be immobilized. So we can do it in multiple different ways. One thing you can do is adsorption to an inorganic carrier. Another thing you could do is trap them in a matrix. So you've got like a grid of things here, and there's, it's all trapped in here. What else you could do is you could have an inorganic carrier and actually covalently bond it. So here's one like weird like carrier looking thing, and here's your thing stuck here. It could either be that it's just adsorbed, meaning it's stuck to the surface, or actually bonded with it. And as well as that, you can encapsulate them as well, like that. So any one of them is fine. So adsorption to an inorganic carrier. Just any way of holding it still, basically, is because that's what immobilization is. Scientists compared the activity of tree tannase and immobilized tannase. They investigated the activity of tannase over a range of pH. So then again, dependent. Well, they're changing. The independent is the pH. The dependent is the enzyme activity. And they're doing this for free tannase and immobilized tannase. Just a real quick pattern we can see is that the activity of immobilized tannase seems to go up, and then levels off, and then starts going down. Whereas free tannase also starts going up and up and up and then down. And you can see that immobilized tannase overall across the board has a higher enzyme activity really than the free tannase, which is interesting to see. So it says calculate how many times more active immobilized tannase is compared to the free tannase at pH 8 and give your answer to appropriate number of significant figures. So what we need to do is go look at what the 8 is and draw a line upwards and then read off the graph. So for free tannase, we've got 0.4, 8, 1.2. And then for immobilized, it's going to be, what's that? That's like 7.5. So how many times fast? It's asking how many times more active it is. You just do 7.5 over 1.2 and then give your answer to whatever significant figures they want. You just follow what the question's given you. So if they've done 2 significant figures, we're going to stick to 2 as well. So 7.5 divided by 1.2, you get an answer of 6.25. 6.25, so roughly 6.3. It allows any answer between, 6.0 to 7.6. So it's 6.8 plus or minus 0.8. Now it says, draw a smooth line of best fit for the free tannase on the graph and answer on the graph. So if we look at the free tannase, if I just remove all this garbage I've just drawn here. So the free tannase, all you got to do is just draw a nice smooth thing connecting the points like that as best as you can. My tablet's really, really messed up, so just pretend I've done it perfectly like that. A student concluded that the optimum pH for this tannase enzyme is pH 5.8. Explain why the data in the graph might not support that conclusion. So what we're going to do is we're going to look at the 5.8 mark, which is about here, and see what's up. So immediately, one of the things I can say is that there are no values between here and here. I don't know if there's just a magic drop there for some reason. Of course, we can assume there isn't, but we should never assume in this sense. Like one would think that, oh, maybe it just will go up and then back down, but like you don't know that until you've experimented and done it yourself. So then immediately I can say there's no measurement between 5.5 to 6 pH. And the idea is, I need to say why that's a problem, because that still doesn't explain why. It's because the optimum could be anywhere between this as well. And that's for free tannase. Whereas for the immobilized tannase, can you see how that range is just huge? The immobilized tannase optimum could be anywhere from 5 to 6.5. It's probably best if we do lots of narrow intervals and really pick out where it is. Other problems that come with this is that there's no indication of repeats, no mention of repeats. The results suggested that immobilized tannase is more stable over a range of pH values than free tannase. Explain why immobilized tannase is more active at pH 8 than free. So if I look at where pH 8 is here, it's way higher than here. Now, the only explanation as to why the free tannase isn't working is that it must have denatured, because pH is going to interrupt its hydrogen ionic bonds and all that jazz, right? So imagine if you've got this enzyme that's fixed down here to a plate, right, versus one that's floating freely. This part can get distorted and mucked up, but this part might be fine because it's fixed and stuck here. It can't actually bend and get weird, right? So if free floating one's like this, and then it becomes all mashed up like this, because that part's gone wrong. Well, if it's fixed here, then it's chilling. So you got to get the idea across that it's probably not denatured as easily, because that's the only reason why the other free tannase must have gone wrong. So it's less easily denatured. We need to explain why. It's because its immobilization supports its tertiary structure, and it kind of stops it from getting all mucked up. Another thing you could say as well is that because this bottom part's here, it might be that the entirety of that side needed to be exposed to the actual pH rather than just, this. So these parts might have been able to be fine, but if we expose that bottom part, it might have bent it. So you could say, the enzyme's not fully exposed to this pH in the solution that they're using. Immobilized enzymes are often used over a greater pH and temperature range than free enzymes. Using immobilized enzymes can be cheaper than using free enzymes. So just two reasons why using an immobilized enzyme in an industrial process could be cheaper. So immediately, one of the things you can say is, imagine like I've got this tube and I'm just throwing an enzyme through it and lots of stuff in it, right? These are just fixed there. So the stuff that comes out is what I want. And I can just keep putting it back in until I eventually get what I need. Whereas if I didn't do that and I had a free thing, I've got to waste a lot more time and money actually separating and taking out the enzymes. So immediately you could say that the product isn't contaminated with enzyme, as well as, so you don't need to extract, no need to extract the enzyme. As well as that, you could say that you can recycle the enzyme, because you could just keep putting stuff in it over and over and over and over and over and over again, and you're fine, because it's staying there. As well as that, they've even told you in the question that you can use them at a greater pH and temperature range, so they might be able to push the temperature higher than a free enzyme would be able to use, and actually make the reaction faster. And those are the things that I would say. Now, it's a state one disadvantage of using immobilized enzymes in industrial processes. So, again, you need to think about what the problems are with doing this. Well, one of them is that it can be expensive to set up initially. As well as that, maybe if you immobilize them, it might actually make the active site shape change and actually not be a good idea. Like, it might actually slow it down a little bit. So you might have to find a compromise between the expenses of separating and a slower reaction. It might be actually faster to use free enzymes, but it might be cheaper to just hold them in place rather than separating them. As well as that, it could be that you can get leakage. So some of them actually just might leak out, So there. Individuals within populations vary. Much of this variation is under genetic control. Two groups of scientists are studying genetic polymorphism in fruit flies. They extracted DNA from two different species of fruit fly A and B. The first group of scientists studied 26 gene loci from species A, and they calculated the genetic polymorphism of A to B, 0.35. The second group of scientists studied the 32 gene loci from species B, and they found that 13 of them were polymorphic. It says calculate the proportion of genetic polymorphic gene loci of species B. So all it's saying is that you've just got to work out the proportion that are polymorphic to the total, right? So that's just going to be 13 over 32, and if we do that, we get 0. Next, it says evaluate the conclusion that species B shows greater genetic polymorphism than A. So we need to talk about, where possible, with every single evaluate question in the universe, supportive statements or data, contradicting, and validity, right? So supportive is what data is directly saying, yes, this thing they're saying is correct. Contradicting is whatever's in the data tables, whatever they've done, saying, no, there's a problem with this. It's directly contradicting what they're saying. And validity is how, can I take this and apply it to every single scenario in the universe? Can everyone else do it and repeat it and do the same things? Are they using an appropriate representation? So for example, if I were to say I did an experiment where I wanted to look at lactose intolerance in people, and I gave them chocolate and they shat themselves. That's not really the best and most direct measure of lactose intolerance, because there's a lot of different types of, lactose and variations of it in chocolate and stuff like that, and the amounts there are. So it's about looking at critiquing every single thing and saying, what could stop me from applying it to absolutely everything in the universe? So supportive statements, if we look already, is that the proportion of polymorphism is 0.35, whereas in B it's 0.41. So the supportive statement immediately is that the proportion of polymorphism in B is 0.42, which is higher than A, so supportive. Now, there's no contradicting statements directly here saying that B is lower than A. Well, there's nothing here in the question that tells you that B doesn't have as much, there's literally nothing contradicting it directly. So all the rest of the issues lie with validity and its issues. So if we look then at what problems there might be with validity, what they've done is they've studied 26 gene loci from species A and then 32 from B. I need to think, is there any way that this could prevent them from saying this conclusion? So, I mean, initially, the first thing that's coming to mind is I don't know how many flies they actually looked at, so no sample size. As well as that, the numbers are quite similar. For example, there's 26, and then they got this to be 0.35, the proportion, right? So if I do 26 times 0.35, I get an answer of 9.1 polymorphic loci, or 9, and 13 here. So the number of actual loci that are polymorphic are quite similar. And then as well as that, there's no statistical test to see if the difference is significant or not, so I can say that as well. And they might have actually done, here again, they're looking at these gene loci, they might not have studied the same loci, because they want to do the same loci to see what these polymorphisms entail. So here's a question on putting the data in a bar chart. I'll just let you guys do that. You put the phenotype on this axis, you would put frequency on this axis, and then you'd put them, with these values basically. So the phenotype would be tongue rolling, non-tongue rolling, so you'd add bars, and you'd do 2 separate bars for male and female. You'd add a key as well, so you could call them striped one and the other one is clear. And then as well as that, you want to fill at least half of the vertical space that they got as well. Since 1940, people have believed that the ability to roll the tongue is caused by a single gene with 2 alleles. So R allows tongue rolling, little r does not allow tongue rolling. The genotype of students who can roll their tongue could be these two. So the results in the table opposite, the total number of students who could roll their tongue was 171. The total number of students who could not was 77. The Hardy-Weinberg principle allows us to estimate the proportion of each genotype. Use this equation to estimate the proportion of heterozygous individuals in this table. So in that case, what we're going to do is we're going to assign what P and Q are. So I'm going to say that P is my dominant, you can choose which one it is, which is big R, and I'm going to say that Q is my little r. And then I'm going to write the genotypes of the possible things here. So people that can roll their tongues, they could be either big R, or big R, little r, and that's 171, whereas the ones that could not roll the tongues have to have 2 recessives, so it's going to be little r, which is 77, right? What I'm going to do is I'm going to work out the proportions or the frequencies of each of these as well. So I'm going to specifically look at U. So little r, the reason is because if that's P is R and Q is R, then I could say, well, that's P, which is the same as P squared. That's going to be PQ, or it could be QP. And can you see how that makes 2PQ? And this here is little r, which is going to be QQ, which is Q squared. So then I know that Q squared is going to equal 77 divided by the total, because we're looking at frequencies. The Hardy-Weinberg equation looks at frequencies. So they all add up to 1, which is 100%. So I'm going to do 77 divided by 171 plus 77. If I do that, we get 0.3105, right? So then that means Q must be the square root of that. So if I do that, we get 0.557. So then if P plus Q equals 1, then, Q has to be 1 minus, has to be 1 minus Q, which could be 1 minus what we just worked out. So if I do that, we then get 0.443, right? So then in this example, P squared plus 2PQ plus Q squared equals 1. They want the heterozygous individuals, which could be PQ or QP, which is your 2PQ. So 2PQ would just equal 2 times 0.557 times 0.443. If we do that, we get 0.4935, roughly 0.494. Let's stick to the same number of significant figures in the question. The Hardy-Weinberg principle might not give an accurate estimation of the proportion of genotypes in the results of the student investigation. The populations of students varies from year to year, and so cannot be said to be stable. State 2 other reasons why it might not be appropriate to use the Hardy-Weinberg principle to estimate allele frequencies in the table. Now, the Hardy-Weinberg principle assumes that between generations, the proportions of these stay the same, right? So if you're doing a calculation and then between generations it starts to change, something is happening to our population. Either it's, for example, you've got things like random mutations happening, or you could have immigration or emigration. And in this case, they don't want to talk about that. So, but then because they've already kind of given it here away that there's, the population is changing. So that's already kind of one given to you. Other things as well is there could be, deaths that are happening prematurely. In this sense, that's not relevant here. It's more to do with squirrels and stuff in a forest or whatever, right? And other things as well is that there's no selection, because obviously if an allele, for example, for tongue rolling made you less likely to survive and reproduce when you're being selected against, then that would decrease. And then as well as that, you just need a big enough population size. So we're looking at 2 to 48 people? That's not really a high number. So the sample size is pretty small. And the other thing as well is they're not subjected to selection either. You're not going to die because you can't roll your tongue before you have the potential to have kids. So mitosis and meiosis are important in the life cycles of organisms. Hydra is a small animal that lives in fresh water. When environmental conditions are favorable, it reproduces asexually. Large numbers of offspring can be produced in this way, and here's the figure for it. So you can see that there's this, and then it's coming off, and it branches off, and boom, there you go. It says asexual reproduction involves mitosis. Name the stages of mitosis. So it's prophase, metaphase, anaphase, telophase. There you go. Suggest why Hydra reproduces asexually when conditions are favorable. Well, imagine you're this thing. You don't really want to start having, like, copies of it when you're not in the right environment. It kind of makes a bit more sense, doesn't it, really? So it's turning it into, it's turning it into what the purpose of it would be for its survival and, continuation. It might be that the moment it's reproduced, the offspring are now happy and chilling in where they want to live and want to survive. And obviously, as well as that, if it's like, yo, this place is quite nice, now I've got to find somebody to mate with. That's going to take up time and energy. So you could say that finding mates takes time. But as well as that, you could talk if this specific Hydra has got specific phenotype that makes it suitable for living in this environment, why not just make exact copies of you so that you can have all of them at the same environment? So because you've got genetically identical offspring, they'll have the same adaptations and also will find the environment favorable. So when conditions are favorable, Hydra reproduces sexually. This often happens in winter. Cells in the body produce sperms and eggs by meiosis. Large numbers of sperms and eggs are released into the water. These sperms can fertilize eggs from different individuals. Each egg forms a tough outer coat and can lie dormant at the bottom of the ocean, the water, until conditions improve. So then it says, explain how sexual reproduction in Hydra leads to genetic variation in the offspring. So we need to talk about things that are causing variation. The reason we have variation is because of alleles being different, right? So then if the allele, like for example, you might have blonde hair or ginger hair. Sorry if you do. No, I'm joking. And I've got black hair, right? That's genetic variation. The reason those have come along is because mutations have led to the changes in these base sequences, and they've just been passed on over time. And as well as that, you're not identical to your parents, nor are you identical to your siblings, because of things that also increase and maximize variation in that sense. Now, from an evolutionary perspective, that's a good thing, because we don't really want all of us to be identical clones, be susceptible to one disease and die. I mean, that happens with like farmers who plant specific veg and fruit, right? If they're susceptible to one infection, it's gone. So we need to talk about the principles that lead to this. So if we do it in order, starting off with like what genetic variation is, how it's come from. So if we talk about from conception, genetic variation is your variety of alleles, which which offspring inherit from parents. The main two factors here are that random fertilization introduces this, and the other one as well is meiosis. Some processes in meiosis increase variation. More specifically, in prophase 1, you've got alleles swapped between sister chromatids in crossing over. So what happens is these are maternal, they're not homologous, these are homologous. Paternal. It's not maternal and paternal here. But what happens is they will cross over and exchange alleles like this. So it might be hair color or whatever, and that increases more possible combinations. So yeah, crossing over. And that changes, you need to explain how that. It changes your base sequence and therefore your alleles. As well as that, you've got independent segregation of homologous chromosomes and chromatids as well. So independent segregation or assortment of chromosomes in metaphase 1 and also metaphase 2 when the chromatids separate, right? So that's talking about it from that sense. We need to, again, look back at this because it's saying in Hydra as well. So in the question, you've got to use what the question's hinted at as well. It's told you that they're releasing large numbers of sperms in water and they can fertilize eggs from different individuals. So that's immediately also introducing even more variation because the sperm can fertilize any egg from different Hydra individuals, which means that it's going to be, for example, it can be carried in water and fertilize eggs from Hydra elsewhere, which, it means even more possibilities, increased possibilities of alleles. That's all it's hinting at. The entire reason that, again, we variate is because of mutations leading to new alleles and variations in that sense. And then meiosis doing these things like crossing over, random segregation, independent segregation, sexual reproduction, having random fertilization of gametes, and also chance as well. All of those things make us very different from each other, and that's a good thing. Mosses are small plants that live in damp conditions. The life cycle of many mosses includes two stages, a gametophyte and a sporophyte. The gametophyte contains haploid cells and produces sperms and eggs. The sporophyte has diploid and produces spores, which can be spread easily throughout the airs. The spores germinate and grow into a gametophyte, and then here's the life cycle. So spores, gametophyte, sperm or egg, zygote, mitosis, sporophyte, and then the cycle repeats. Cool. The zygote grows into the sporophyte by mitosis. The haploid gametophyte of one species contains 28 chromosomes. A single DNA molecule has 2 strands. Calculate the number of strands present in the nucleus immediately before mitosis. So there's a multiple step thing going on here. What we've got is we've got 28 chromosomes, but this is a haploid thing. So a diploid one that's chilling is going to actually be 2 times 28. But then right before mitosis, remember you've got DNA replication as well. So you've got haploid, which will fuse together to make a 2 times 28, but then in DNA replication, those are doubled again, right? So you're chilling with, if we were to pretend your chromosomes were already expanded like this, a diploid organism will have maternal and paternal. Right before meiosis, what will happen is they will make doubles like this, which is where those sister chromatids come from, where that's maternal, maternal, paternal, paternal. So in that case, the answer is going to be 2 times 28 times 2, so 2 times 28 times 2, right? Which would give you 112 chromosomes, but the question's asking you how many strands. So each one of these is 2 strands. So it's actually, the strands is going to be that doubled as well. So 2 times 112, which gives you 224. So it says mark X on a figure at which point meiosis occurs. So if we have a look, we're looking for where it becomes haploid, right? So here the question's told you that these, you produce spores, which can spread through the air, and then they could become, they can germinate and become a gametophyte, right? And gametophytes contain haploid cells and produce them. So basically it's any point between sporophytes and spores, really. Because at that point, between here and here, that's when they're becoming haploid. So the diagram of a moss sperm is shown. So it's got a nucleus, it's got this, and it's a flagella. The flagella allows a sperm to move towards an egg, suggesting and explain the other adaptation that's likely to be present in this. So, I mean, there's multiple things you can say, and obviously it's similar to moving towards an egg. It has to break in, so you could say it has an acrosome, which has enzymes to penetrate the egg. You can't say it breaks the egg cell wall, because it's not a plant, it doesn't have that. It's moving actively, so that means it's going to require energy, which means it's going to need more mitochondria, isn't it? So many mitochondria, so there's more ATP for movement. You can't just say what it is, you need to say why as well, and there you go. Carbon and nitrogen elements that can be recycled. Here's the flow chart, and it says identify the processes at A and F. So if you look at A, we've got fossil fuels, and we become CO2 in the atmosphere, so that one is combustion. And we're looking at F, so we've got producers going to CO2 in the atmosphere. So producers are making CO2, that's got to be respiration. The concentration of carbon dioxide in the atmosphere varies depending on the time of year, suggesting why the concentration in the atmosphere increases during the winter months and decreases during the summer. So, I mean, there's 2 reasons. One, in the summer, there's more light. In the winter, there's less. So if there's less light, there's going to be less photosynthesis. But as well as that, in the winter, we are cold and we want to stay warm. So we're probably going to burn more fossil fuels to stay warm as well. So either one of them is fine. In plants, the glucose produced is changed into starch for storage. Glucose and starch are both carbohydrates. Show the main differences in the structure between glucose and starch. So the differences, well, one of them immediately is that glucose is a monomer, starch is a polymer, right? Then as well as that, you can go ahead and say, well, I mean, glucose is a monosaccharide versus a polysaccharide. Glucose has no glycosidic bonds, whereas starch has glycosidic bonds. I'm too lazy to write the whole thing. You obviously write the whole thing. As well as that, here, there's in the formula glucose is C6H12O6 has more H's and O's, whereas in starch it's got C6H10O5 because it's lost an H during a condensation reaction. So there's less H and O due to condensation, whereas this has more H and O. So any of them is fine. Carbohydrates only contain the elements carbon, hydrogen, oxygen. Polypeptides contain carbon, hydrogen, oxygen, and nitrogen. What's something else that would be in polypeptides? Sulfur, because they make disulfide bridges. The nitrogen cycle shares many similarities with the carbon cycle. Describe and explain the similarities between the nitrogen cycle and the carbon cycle. So in the nitrogen cycle, real quick crash course, what happens is you've got atmospheric nitrogen, which then gets fixed into ammonia, right? Which then becomes ammonium ions when dissolved, and then it can get nitrified into nitrites with nitrates, and then it can get denitrified and returned back to the atmosphere. As well as that, those nitrates are taken up by plants, which are then scranned by animals, by feeding, and then those animals die, the plants die, and they get broken down by saprobionts, and they release ammonia, and the cycle repeats like that. So in a similar sense, the carbon cycle follows the same principle. So what you want to do is just go step by step to see what's, going on. So in this case, in the carbon cycle, we have atmospheric CO2, which is then converted into something that can be used by other organisms, right? In this case, it's being, CO2 is fixed into, glucose and useful stuff like that in photosynthesis. So immediately we can say that. Whoops. It's only looking at similarities. So one of the similarities is that atmospheric CO2 and N2 are fixed into compounds like glucose in photosynthesis and proteins from ammonia via nitrogen fixation, right? And if we look, these plants are then scranned by consumers, which then die and put stuff out in the same way I showed you before, that they scran and die, right? So they both have the element of that. So the producers in both are eaten by consumers, and both of those die and decompose, the producers and the consumers, decompose by, saprobionts and other macroorganisms. And what they do is they release CO2 again back into the atmosphere, right? And they also release the N2 back as well, right? Well, N2, I should really say NH4 plus, right? And in both of them, you can eventually return it back to the atmosphere. I mean, both can return CO2 and N2 to atmosphere by decomposition for CO2 and denitrifying bacteria for N2. Those are some pretty hefty similarities that I would say as well. And the whole idea is that you're getting across that both of them involve getting atmospheric CO2 and N2, putting it into compounds, things eat it, digest it, break down, return it to soil, and so forth, and the cycle repeats. Algae are photosynthetic organisms that live in water. A rapid increase is known as algal bloom. Scientists study the population of algae in a river at different times of year, and the results are shown here. So again, dependent what they're changing, the time of year. Dependent is your algal population, right? In cells per centimeter cubed. It's just a quick idea of what's going on. From March to May, it goes up and levels out in April and then goes down. And then from May to June, it goes, June, July-ish, it goes back up and then down again. And then July to August, it peaks and then goes back down. You can see there's just a repeating pattern of up and down and up and down, up and down, right? It says here, calculate the percentage decrease in the population of algae between the peak population and 1st of November. So if the peak population is the highest it'll ever get, which is here. So that's about what, 21500. So the peak population is 215000, and then let's find the lowest. And then it's asking us for what the 1st of November is, which is about what, like 20,000. So November is 20,000. Your percentage change is always your second. So percentage change is going to equal 100 times the second thing, so the after, minus the original over the original. If we do that, we get minus 90.697, which is roughly minus 90.7. That shows that there's a decrease here. So then, in that case, the answer is 90 points. So they want it to, how many significant figures they give us the question? They've given us 2 significant figures, as you can see, so we'll stick to 2. So it's going to be 91%. You can put the minus in. The minus is showing that it's going down, but, there you go. The river in which the city was conducted is described as a dynamic ecosystem. Use this figure to explain why it could be described as dynamic. The idea is that, I was just saying before, it goes up and down, up and down, up and down, up and down, right? So it's showing that there is, it's changing over time. So it's over time, it's changing and, increasing and decreasing. And there's some numbers as well. So from 1st of March, it was like what, 10,000, then peaked at 60, is that 60 cents? Sorry, 70,000 in April, then decreased to 30,000 in mid-May. Then it peaked up again to somewhere else, just showing them I would say 2 cycles. Then it goes up to June again to this and then back down, showing that it's going up and down, it's fluctuating. A student concluded that the increase in the population of algae was due to higher temperature and higher light density in the summer months. Considering this as the student's data source, discuss the weaknesses in their conclusion. So we're looking at problems with this now. So we're not looking at supportive, we're looking at what's contradicting it and issues with validity. So in this case, let's look at some problems with validity. So validity again means, can I look at this and directly say this is going to give us our conclusion? Is it an appropriate thing to say? Well, immediately, number one, we have only up to November. There's no data on the winter months. So no data on winter months, which are December, January, and Feb. As well as that, they're only looking at the month and the algal population. Lots of other things change in these times as well. So there's no data on temperature or light, which can be a problem if they're trying to conclude that this means that there's going to be more light and higher temperatures in these months because there's no data on it. As well as that, surely if we were looking at the summer and there was a good amount of salt, it should just sun, it should be fine, but it's actually going down. So there's a decrease in summer months. So then it must be that, well, we need to think about other factors that can affect this. We can't just say other factors. I really think you should go ahead and give an example. Other factors may contribute, like, competition, for example, or increased nutrient availability or decreased in predators. As well as that, you could say that there's no correlation does not mean that there's a cause, correlation does not imply that there's definitely, a pattern between this going up and this going up, and therefore this going up and this going up, just because these summer months are correlated with temperature and light, and these are correlated with the months. You can't say that either. I think you can say the other factors is always, if you're going to say other factors, you got to say what the factor is. I just don't think it's appropriate to just go ahead and say other factors, because a lot of mark schemes just don't like that. So many algal species are unicellular organisms. Some occur in colonies of more than one cell. Here's a typical cell. What have we got going on here? It says, at various times the algae have been collected and classified in different kingdoms. Use the information above, draw a conclusion at which most appropriately classifies them, and in your answer explain why other ones are not appropriate choices. So in this case, what we have going on is it's got mitochondria, it's got thylakoids, it's got a cell wall made of cellulose. So I mean, the mitochondria, the nucleus, and the thylakoids as well, those all are double membraned organisms, which mean it kind of fits more of the eukaryotic side of things. But that being said, it's unicellular, so it's not really going to be like a plant A. And also, as well as that, it's got a cell wall, so that kind of makes it more plants. It's all over the place, isn't it, really? But then as well as that, it's got a cellulose cell wall, so it's not going to be fungi, which has more to do with chitin. So the only thing that really fits this awkward in-betweeny thing is Protoctista. And that's exactly what, we'll just write exactly what we said there. So Protoctista, because it has double or membrane bound, either one's fine, I'm going to say membrane bound, organelles are present, e.g. the nucleus. So it's eukaryotic, not prokaryotic. You have to say what that significance means, what it means. As well as that, it's unicellular, so not plant A. But then as well as that, it has a cellulose cell wall, so it's not fungus or fungi. And as well as that, it has a cell wall, so it's not part of Animalia. Right? So you've got to go and look at these in that sense and sort of look at the features that make these and say why it's this and not that, why it's this and not that. The human body is able to protect itself from disease in a variety of ways. Here's a lot of cells and structures. Which letter or letter indicates the structures involved in preventing the entry of pathogens in the body? So when we're looking at stopping things getting in the body, what we're trying to do is we're trying to stop them from getting somewhere else. Like, for example, the goblet cells. Goblet cells are in the airways and they make mucus, which traps like dust particles and microorganisms and stuff, and it stops them getting to the alveoli. So that's definitely one of them immediately. Other things as well, some mucous membranes as well. They're another one because they'll have like enzymes and stuff that stop the things getting digested and broken down and this and that. So that's immediately one. As well as that, what else have we got? We've got platelets. So platelets clot and can clump and block things out of the way and stuff. So that's immediately one. And J, it's a skin. Skin is the skin barrier as well. All it's trying to show you is how we're stopping and getting from the place it wants to be. So, like, for example, your skin, right? It's a barrier. It's a physical barrier. As well as that, those mucous membranes also have things like mucus and enzymes and lysozymes and stuff like that. And it can also have phagocytes, which can get rid of them. As well as that, things like tears and urine also have lysozymes and also acid in the stomach. We also have things like coughing and sneezing, which are reflexes to expel, remove things, diarrhea as well, and vomiting. And as well as that, things like clots and thrombi as well kind of block things out. So all of these are to do with the fact we're trying to stop the pathogen getting to where it is. Here then, it says, which of these letters or cells are physical barriers to the entry of pathogens? Well, I've just said two of them already, so it's going to be I and J platelets, because they stop physically. Then it says, which letter or letters indicates the cells or structures are involved in phagocytosis? So phagocytosis will involve, let's have a look at the options again. So phagocytosis, well, I mean, it involves antigen-presenting cells, because they themselves act as it, and other ones can present to them and make them do that as well, so that's immediately one of them. So we've definitely got A. Erythrocytes are red blood cells, goblet cells, no. D, lymphocytes are no, they're more to do, this is involved in phagocytosis. E, well, lysosomes fuse the phagocytic vacuole. Remember when it surrounds it in the pathogen and holds it here, and then, it surrounds it like that, and then it puts a phagocytic vacuole like this and releases lysozymes into there. So lysosomes fuse with it, sorry. So that's immediately one as well, so that's E. Neutrophils is definitely one of them. They're a type of granulocyte which do phagocytosis. And then as well as that, H, phagosomes as well, which is the thing that we're saying there. Phagocytic vacuole, phagosome, same thing. Then it says, which letter or letters indicates a tissue, and explain your answer. So if we have a look, cells, organelle, membranes, that's a whole bunch of cells, so that's a tissue, organelle, cells, and organ. So remember that tissues are all about different cells working together, or a few types of cells working together to form a function. So F, and that's for the exact reason, is that it's a few types of cells performing a function. So phagocytosis involves cytokines and opsonins. State the role of cytokines and opsonins in phagocytosis. So cytokine, cyto means cell, kine is movement. So they are chemical signals that are causing cell movement. So what they do is they attract phagocytes. Opsonin comes from the Greek word opsonia, I don't know how you pronounce that, but it basically means to make something tasty. I'm not joking, I'm dead serious. So if it's something that's tasty, and phagocytes, phago means eating, cytocell, eating cell, it's making it tasty for the phagocyte. So what it does is it binds to pathogens, which is going to be the thing that's going to be eating it, to increase phagocytosis. It's literally marketed being like, hey bro, look at this tasty snack, and it's like, yum yum. Chickenpox is a common disease. People who have recently recovered from chickenpox can donate plasma so that their antibodies can be given to leukemia patients with weakened immune systems. Use a tick to indicate which type of immunity is functioning in the leukemia patient when given this. So in this case, it is all artificial because we're injecting these antibodies, and it's passive because they're not making their own antibodies. They're not undergoing the primary immune response. And we'll explain your answer there. It's because it's injected, so that's the artificial part, and the patient is not producing their own antibodies from a primary immune response. Thank you very much. I hope that helped. Please like, share, and subscribe. You know the drill. And yeah, see you next time.